just to point out that this one says, due to cancellation, there is a slot available at St. John's tonight. If anyone who isn't going would like to go and talk to the reception desk. And to remind you, please don't run off to lunch, because we're going to have our group photo taken just outside, straight afterwards. Okay, so I'll hand over to JP Morgan to chair the session.
Nice, welcome everyone. We're delighted to have one of our organizers in the special plenary session, Rosemary Bailey. And Rosemary tells me that included in her talk are going to be things that she's thought about and worked on over the past few weeks of the workshop. Certainly in the spirit of everything that's been done here. We'll be delighted to hear that and everything that you have to say, Rosemary.
Thank you. And I want to begin by once again thanking Dave Woods and Heiko Grossman. They have really organized the scientific program for this week. And I haven't been an organizer for this week at all. I'm having an absolutely fantastic time. Thank you to both of you. So I'm going to say some things about the design of two-phase experiments. What do I mean by a two-phase experiment? Well, what I've thought about first of all, but I've put the typically in brackets, is you have some phase where you apply treatments to experimental units, and then you take the produce, whatever it may be, from those experimental units and allocate them to a different sort of experimental unit for some other processing or for some evaluation. And so the way you analyze the data at the end will depend upon the design at each phase and the way those designs fit together. Well, usually, I mean, this would apply in any design, you want to estimate the most important contrast with low variance and, if possible, a large number of degrees of freedom for the appropriate residual. And you find that if you've got a two-phase experiment, those two criteria can conflict. So really, I'm going to be discussing issues and examples. There won't be very much in the way of formulas. And there's Desmond Patterson's design key was a tool I learnt a long time ago, and I found it can be quite helpful for thinking about how to construct two-phase experiments. Some of the stuff I'm reporting here is based on ongoing work with Chris Bryan. The examples, you can see there are a list of people that I have talked to. Some of the examples I'll give you are sort of real and some are generic, based a sort of compound of lots of things I've talked about. And there's also been discussions with Qing-Chui Sheng about some of the issues. So my first example is one that was presented at the Joint Statistical Meetings in New York some years ago by T.B. Bailey, who is no relation of mine. And in the first phase, they're making meatloaves. I could be present or not. And there were 3 levels of irradiation. And this was done in an absolutely classic way, 3 blocks, 3 loaves per block, randomized complete block design. Then at the second phase, in each of 3 sessions, there were some panelists and time orders making this number of tastings. So portions from these meatloaves were put into this. Actually, this indicates an orthogonal design. It was a pair of Latin squares. So in the first phase, I start off with a systematic design. I randomize it by permuting blocks, permuting loaves within each block. Now, in the first talk, when the conference began, David Cox talked about randomization and how that might link with models. If you have a model which is defined by that randomization, this means you get some variance term everywhere, some covariance for loaves within the same block, and possibly another covariance for things in different blocks. Well, I would usually reparameterize this in this way. This is a variance term within blocks. This is one between blocks, and I've possibly got something else for the grand mean. I write it in that form when I'm simply using these Q's to take up less space. These are what I would normally call the stratum variances, and the point is that these things have to be positive because they're eigenvalues of the variance covariance matrix. And these Q's are mutually orthogonal idempotent. So we always get something in that form. That's from the first phase, and these would be 18 by 18 matrices. In the second phase, the systematic design used here was a pair of 6 by 6 Latin squares. What would you do? Randomly permute sessions, panelists within sessions, time orders within sessions. I won't go through the whole thing, but that gives you a covariance model, again in spectral form. I'm starting at the other end now, something on the grand mean, between sessions, between panelists within sessions, between time orders within sessions, and everything else. So when you put all that together, oops, this is the one I've just developed, and here we have more or less what I had before, but I've had to multiply by 12 because that's the replication at the second stage. And these Q's I've put a tilde on because I've had to spread these matrices out over 216. So we get a covariance matrix that can be expressed in that form. So now I think about what I call a skeleton ANOVA and what Chris Bryan tends to call a decomposition table. I start off with this decomposition of the tastings tier. So those are the items I've just talked about, and I put with them the corresponding variance parameters. Now I look at the middle one and I associate these terms with the appropriate things there. So of course the mean always goes with the mean. What happens to blocks? Well, we've aliased blocks with sessions. Then what happens to the rest? We've deliberately made the rest of it orthogonal to panelists and time orders, so the rest comes right down there. Now 15 is less than 165, so you get some residual there. Then what do I put in? For each of these terms, I have a corresponding variance component. And those are simply added to the ones we had before. Now I go over to the left and put these ones in. Well, again, the mean goes with the mean. These are confounded with loaves within blocks down there. And so here is the 1 degree of freedom for adding Rosemary or not, and we have to add a quadratic form in the differences of the effects. Then similarly for irradiation, similarly for the interaction, and 5 is less than 15, you get that residual there. One of the main points that my namesake was making when he presented this example is that the appropriate residual for testing these has only 10 degrees of freedom. That is the residual mean square, where the variance components are what you would get here if you haven't got any treatment effect. And so he was banging on about the fact that quite a lot of people would analyze this wrongly. They would combine those residuals. So there's two things here, quite small residual degrees of freedom, but we have managed to pick up the smallest, we assume that will be the smallest variance component from there, and that would be the smallest one from there. So I'm sorry if this sounds a bit too schoolmistressy. I couldn't think of a better word than lesson for this. But this is really, as I say, T.B. Bailey's lesson. If you apply some treatments in phase 1, the number of degrees of freedom for the relevant residual can never be increased by what you do at phase 2. You can make it smaller. You can do some silly things and make it smaller. You can never increase it. So I think that gives me, I'm trying to get some guiding principles. And here we had treatments orthogonal to the large blocks in phase 1. And quite a good way of making sure you don't lose any more degrees of freedom is saying if we've also got some sorts of large blocks in phase 2, then confound the two sorts of large blocks with each other. You're not interested in either of them, so confound them with each other. Don't waste degrees of freedom. Here's another example taken from a paper by Wood, Williams and Speed, and just before Terry starts shouting at me, I have to admit that they used this as an illustrative example. It wasn't exactly a real experiment. It starts off by looking just the same. It's on storing milk, 3 temperatures at which it could be stored, and 2 types of container. And by adjusting a real experiment, they said, supposing we actually had 2 replicates and 6 milk volumes. You may notice when I do these things, I sometimes use awkward words. It's because I deliberately try to make sure that all the first letters are different so that I can abbreviate. So this is why I call these milks. So 6 milk quantities within replicates. And so this starts off very similar to the other one. At the second stage, there were 2 sessions, 8 tasters, but I can't use T, so I've called them judges. And in each session, each taster tasted 6 drinks, so obviously it could be one of each of these. Now, according to the principle I've just given you, treatments are orthogonal to this replicates thing. We should confound replicates with something we're not interested in over here. We might confound them with sessions or with judges, or we could even do some Latin squares up there. So if I followed the principle I'd just given you, I would do something like this, confound replicates with sessions, and then we would get an analysis which I won't go through because it's very similar to the one I showed you before for T.B. Bailey's example. So the interesting thing comes down here, the containers, the temperatures and their interaction, and that residual. So they're confounded with the smallest thing here and also the smallest thing here. And again, so we can't increase that number of residual degrees of freedom, but we've done our best to make the appropriate variance small. Well, what did they actually do in the paper? Remember, Terry? What they did in the paper is this spider. This notation means their design was so elegant and complicated that the only way I could show it to you would be to actually write it out in full detail, which I'm not going to do. And it ended up, I've worked out the ANOVA table in the sort of form I'm talking about, and I'm not going to talk about all of it or how I got it, but I'm going to point out some differences. First of all, instead of saying replicates are boring, we should confound them with something boring, they've put them down here, which is potentially using up one of these degrees of freedom. The second thing they did, oh, sorry, I should have done that, was this 10 degrees of freedom for the milk volumes within replicates. Their design actually had to split that up into 2 parts. The 2 parts, one of those parts was partially confounded in sessions by judges, partially down here. And the efficiency factors were 1/3 there and 2/3 there. And when you've got this partial confounding, these efficiency factors then turn up as multipliers in the expected mean squares. As a result of doing all this complicated stuff, although the residual down here, we've still got the 2 smallest variance components, but looking at it just as straightforward ANOVA, some residual degrees of freedom have been lost. There's only the 2 there. Now, to be perfectly fair, the purpose of this paper was not to say this was a good design, it was to say how you might do all these complicated things. But I'm saying I wouldn't design it like that. So, that was my first point. The principle is, if you've got some large blocks in the first phase, and your treatments are orthogonal to those large blocks, you've got some large blocks at the second phase, it's probably a good idea to confound the 2 together, and you also want to watch out for losing degrees of freedom. So now on to a second thing to think about. Earlier in the week, Steve Gilmore was talking about some factors could be hard to set. Well, in the industrial context, he's thinking of factors where you've got to go and turn lots of dials to set them. With my more agricultural background, I think about things that can only be applied to a large area of a field rather than a small area. So if we've got one of these hard to set factors, probably in the first phase, it's in a stratum with a larger variance. Now, we've already seen, I mean, I haven't proved it, I've just shown you that you have to add the variances from the two phases. So if you've already got a large variance from the first phase, what should you do at the second phase? Well, I think that because you've already got a big variance from the first phase, if at all possible, you should try and put that at the second phase in something that has a small variance. So, because in the first phase such a factor would get a big variance, you don't want to continue to add in more big variances from the second phase. So, this is an example. This is something joint with Cathy Ruggiero. Now, the numbers in this example are exactly right. It is a real experiment, but some of the details I'm being a bit vague about because it's an experiment that a PhD student is currently running, and I don't want to, spoil him getting a PhD and getting a paper out of it. So, there are two things which I will just call interventions, and although they're applied to animals, it's only feasible to apply them to whole cages. So I've got 8 cages, 2 animals per cage. We're also interested in 2 different tissue types which are taken from each animal. At the second stage, they can do 8 runs, and she actually called these things 8 different colours. I've called them labels just by my using different letters principle. And this question mark means this is Cathy talking to me and saying, what should we do at the next stage? Well, what should we do? What should we think about? These interventions in the first phase is confounded with cages. It probably has the biggest variance. So at the second stage, we probably want to confound it with a low variance term. And for me, that means probably making it not confounded with runs, not confounded with labels, but orthogonal to both. We don't want to lose any degrees of freedom here. So if we're making whatever we do make cages confounded with, we'd like the whole of cages to be confounded with. So that's thinking about the interventions. We've also got to think about the tissue types and the interaction, and really, if possible, we would like those also to be orthogonal to runs and to labels. And this is, it was actually an example of Andrew Mead's that made me realise that the design key could be useful, so I already had that in my mind, and I'm going to show you how we used it in this case. We express everything in terms of 2-level factors or pseudo-factors. So this is easy. I and T both have 2 levels. 8 cages, so we have 3 2-level pseudo-factors, C1, C2, C3. Another pseudo-factor for animals in cages, another for positions in that. 8 runs, 8 labels, we deal with them just as we did with cages. So now how do we go about? The design key says we have to choose aliasing between there, and then between there and there to achieve what we want. So the interventions have to be applied to cages, so I can alias I with actually any one of those, or indeed any combination of them. And I would say here, this is where I would put on my pure mathematician's hat and say, without loss of generality, I can equate I with C1 and not mess around. But of course, there's actually 7 possibilities there. And if you're doing a computer search, things can blow up. Well, then what do we want? We said that cages shouldn't be aliased with runs or with labels. So we want each of these C's to be aliased with some combination of an R and an L. And we want to do that in such a way that if we take any combination of C's, it's also aliased with a combination of R's and L's. Well, there's a dead easy way to do that. You simply say C1 is R1 plus L1, C2 is R2 plus L2, C3 is R3 plus L3, and then you'll see whenever you take a combination of those, you've got R's and L's in it. What about A? Well, A is only animals within cages. So A and all combinations of A with any C's give you the rest of animals within cages. That's 16 animals. You can't make 16 orthogonal to 8 runs in something of this size. So something within there will be aliased with runs, and there's really, again, no loss of generality in taking A itself to be one of those R's. Because if you don't take A to be that, some combination of an A and a C. When we get down to P, well, everything with some sort of P in it accounts for positions, and the same sort of without loss of generality. I've chosen L2. I couldn't choose L1, because if I'd chosen L1, I'd have some linear dependence in there. But those bits are really quite straightforward. Forced, easy, they're all without loss of generality, but there's nothing difficult. Now, this is the only place where you really have to apply your brain. What should we alias T with? Well, it has to be something with positions. And I say positions are everything that has a P in. So it's got to be P plus, well, if we had P on its own, we've already put P with L2, and T would then be aliased with labels, and we don't want that. So it's got to be P with something from here, and you try a few things. You don't have to try very many, and I found that P plus C3 will work. And I'll show you on the next slide why I know that works. So that's just our picture with the panels again. This is the aliasing that I've given you. This is showing you how you do some of the calculations. First of all, something with no treatments in. If I take P plus C2, that's L2 plus R2 plus L2. These are all mod 2, and the L's cancel out. I just get R2. This single degree of freedom comes from the variability of positions within animals and cages. This single degree of freedom comes from runs. So when I start to get the ANOVA table, I will have a component from that variance and a component from that variance. Let's look at the treatment ones, because those are important. I've aliased T with P plus C3. P is then aliased with L2. C3 is aliased with R3 plus L3. Nothing cancels. We've got some R's and some L's. So when I do the same thing, what have I got there? We'll have something for tissue differences. We've got the variability for positions within animals and cages, which should be the smallest variability from here. And we've got the variability for interaction of runs and labels, and that should be the smallest variability there. Well, we still need to care about this interaction, so when I thought this was possible, I had to then go and check this as well. I is aliased with C1, T is aliased with P plus C3, so this is what we get in the middle. Now how does that go on? C1 becomes R1 plus L1, P becomes L2, C3 becomes R3 plus L3, and you'll see that that doesn't cancel out, so we really have got RL interaction there. So the interaction between the interventions and tissue types, the variability that it gets comes from the smallest one here and also the smallest one there. well, I probably won't go through the whole of this table with you, just the bits that I've really talked about. The interventions had to be aliased with cages. But we made sure then that cages were aliased with the stratum here that we hoped would have the lowest variance. So although we've got the big term there, the eta C, we've put it together with the psi RL, which we hope should be smaller. Also, we could never increase, we could never increase that residual degrees of freedom, but at least we haven't decreased it by putting the whole of cages in there. The other two terms, the tissues and the interaction, we've managed to get the smallest variance from the middle and the smallest variance from the last phase. So I think I've followed the principles as far as I could, and Kathy was actually perfectly happy with that as a design. Now I'm going to just draw your attention to some other odd things. Look up here. If you calculate, you can actually calculate all of these. If you need to, you simply write down every single degree of freedom, find out its aliasing, and that tells you what you get from there, what you get from here, what you get from here. Here are three, I'm not sure whether to call them strata, they're certainly subspaces, where we have got the runs variance coming in, and we've got the animals within cages and the positions within that. So we could equate those mean squares to their expectation and get nice linear estimators for those etas and that psi. Well, now look down at the next three. Similar, but with lines. So we could get nice linear estimators for these three things again. Aha, but those two are what we already estimated up there. Well, same story again down here. So if we just go about this in a rather simple-minded ANOVA way, if we just test that against that, and these against those, we're not using some information we could have got from up there about the sizes of those variances. So I'm going to come back to that in just a minute. So what do I hope I've got from that example? In both phases there, the design was orthogonal. I used the design key. I think that's a simple method of constructing the design, and it's also quite simple to find out what's confounded with what. As I say, you just simply write down everything you're interested in, then say what's it aliased with at the next stage, what's it aliased with at the next stage, and so on. Second thing, I hope you'll see, by looking at the skeleton ANOVA table, we see the expected mean squares for what we're interested in, and also for the residual terms. We see the residual degrees of freedom, and so I think that's a very useful tool for comparing designs. Write out those tables, what's got higher variances, what's got higher residual degrees of freedom, and so on. But I did show that if you just go about it in a simple-minded way, equating mean squares to their expectations, you may get some different estimators for those variance parameters, and of course they may not give you the same result. So how do you analyze? I don't know. I really don't know. This is a question that I hope some people... Okay, Terry, you can jump up and say throw it into REML, so I'm going to... quotation, right? You might think about doing ANOVA just by looking at the sort of table I was showing you. And Chris Bryan and Roger Payne have a procedure in GenStat that does precisely that. But that might be, losing some information in the sense that there is other information in what those variance components are. Now, I'm not sure if these two should really go together. I mean various ways of estimating the variance components and then doing generalized least squares. Now, that was quite a complicated table. Some of the degrees of freedom were rather small. The justification for REML is that it's asymptotically correct. 1 and 2 degrees of freedom are not asymptotic in my book. And indeed, Steve Gilmore and Peter Hoos have done some very interesting work showing that when you've got small degrees of freedom, REML, and in fact several of the major packages, can give you really silly results. Now, I don't know if that applies to an orthogonal design or just to non-orthogonal ones. So you might think of, just use your favorite mixed model software. Well, Chris Bryan has done some experiments, isn't quite the right word, but he's tried out some of them. And it may not be able to cope with the sort of confounding that's going on. So what do I mean? In the particular example, there was a single degree of freedom for this pseudo factor R1. R1, so you know that comes from runs. But we also made it to be confounded with animals within cages. So when you look at that in the table, you get a variance term from runs, and you get a variance term from animals. The way you specify to your mixed model software, we've seen the sort of formulas that Chris was writing down. You tend to give a list of terms in an order. And my test for is your program doing something sensible is if I write down runs before I write down animals, and if I then do it again writing down animals before I write down runs, do I get the same answer? Because I think that most of this software did not envisage having some degrees of freedom which incorporated variance components that both turn up in their own elsewhere in the table. So I really don't know the answers to this, and I would like to know. Okay, on to another. Now this example is more generic than actual from lots of conversations with Ruth Butler, who is now with Crop and Food in New Zealand. So we're going to just compare 27 varieties. Probably this is a grain crop out in a field experiment, and later on we're going to do something in the lab. Now when I was at Rothamsted, you would take your tractor and do your ploughing in some direction. Then you would do your sowing in the same direction. If you're going to go along and apply something, a spray, you would do it in the same direction. And when you harvest, you do it in the same direction. Now I was horrified to find out from Dave Saville in New Zealand that they don't always do it like that there. They do some operations like that, and they do some operations in the other way. Yeah, okay. You can shake your head, but it's what they do. So that means in some sense they have to think about rows or big rows and big columns in their fields, whereas in Rothamsted we could get away with thinking of blocking in only one direction. So we've got 27 varieties, 3 rows by 3 columns. You obviously can't make that orthogonal to all of that. So I'm going to go straight for design key. These now all have 3 levels, and you can do something like this. Something has to be confounded with something in rows and columns. So make V1 and V2 aliased with those plot pseudo factors, but V3 has to be confounded. Now this is not a hard to set factor, it's something forced by the fact that the row column combinations aren't big enough to put all the varieties in. We're doing an incomplete block design. How do you design this next phase? Where, actually this is, so it's illustrative. We've only got the same number of things here as there. How are we going to group this? We get the produce, we put some of it in little bottles or whatever. How are we going to put it into batches for the chemical analysis? Well, one possibility, and this is probably the one that's most obvious, is we've got these 9 row column combinations and we turn them into the batches. Now I'm not going to show you the whole ANOVA table, I'm just going to show you what happens in the batches stratum. So here we've got the rows and the columns and their interaction. This V3 we confounded with that, so it's there. Now, its residual is only 2. That's horrible, but there's actually nothing we can do about that because that was forced from the first phase. What is even more horrible to my mind is we've got this large variance component from the first phase, and we have quite deliberately added on this big variance component from the second phase. So you've got the biggest possible variance there with hardly any residual degrees of freedom. That's horrible. Let's not do that. So we'll try something else. Just having rows aliased with batches and one of these plot factors. So again, you work it all out. So now what happens in batches? 2 degrees of freedom for rows and 6 for the plots within rows. And now, because we've got P1, which is aliased with V1, we've had to sacrifice some information on varieties. Given the numbers, you've got to sacrifice something, but I'm suggesting it may be better to sacrifice a different part. So, although it's got this big term there, it goes with that smaller term. Residual degrees of freedom are now 4, not ideal. I can actually do a bit better than that if I manage to make sure that the things confounded with batches, I've done it in a slightly more cunning way, I don't get any R or C terms in, so that everything confounded with batches comes from the plots within rows and columns. So I've still sacrificed this, but this is with the smaller variance there and the bigger variance there, so that's better than the other one that had all the big variances. And this now has residual degrees of freedom 6, which you can see is the maximum you could hope for, given that you've got to put something in this stratum. So I would say that under the constraints we've got here, this is what I would recommend. So what have I done? I first of all said we've got to confound something here. I then said, given that, we don't want to lose information on that at the next stage. I tried some possibilities in here, and again there's an awful lot of without loss of generality on the type of things that you try. I didn't like the one where I lost even more information on that. You have to sacrifice something. I've chosen to sacrifice that. Given that, I tried to make as many residual degrees of freedom as possible go with that. So, this is a principle I wrote down before for a hard to set factor, but it might be something that's hard to set. It might just be that because your blocks aren't big enough, you've got something confounded with blocks. So if in the first phase there's a treatment term that has a large variance, then I think what you ought to try and do is allocate that to a phase 2 stratum with small variance, so you avoid this thing of adding 2 large variances. And in the second example, we found that this actually forced us to sacrifice some information on another treatment term. And of course, it might not actually be practicable. It depends on the relationship between the 2 phases. Do you have to take a whole row from the first phase and process it before you go to another row? Or can you put the stuff all into little bottles and then rearrange it? So you need to think about both of those. Well, all the designs I've looked at so far are, well, apart from the Wood, Williams and Speed one that I didn't do in a lot of detail, the ones that I've really gone on about have been orthogonal. Something is nicely in a stratum or it's not. Supposing we've got a non-orthogonal design at the first phase, supposing it's got information in more than one stratum. Well, the principles I've given you so far seem to conflict with each other. What should we do? So here's an example. This is a made-up example, but it is based on lots of conversations with various people about real examples. Well, after yesterday, you all know about two-color microarrays. Where are we getting the stuff from? We're getting the stuff here from 12 animals, so we'll have 2 technical replicates of everything. The complication is that the animals come in litters. And at the first stage, I've used this word treatments in a loose sense. It might actually be properties that we had before, but no, let's, I think, have it as treatments that we can allocate. First phase design. 6 treatments, only 3 animals per litter. You've obviously got to have an incomplete block design, and in most senses of optimality, I tend to use A-optimality. This one is A-optimal. This particular design occurred on one of Heiko's slides at the end yesterday. There's obviously some nice things about it. This forces you to think of the treatments in 3 groups of size 2. This might be artificial, it might not be real, and you'll see that every litter has one thing from each group. So groups are orthogonal to litters, but comparisons like 1 versus 6 are not orthogonal to litters, and you can show there's an efficiency factor of 1/3 there. Second stage, 12 slides, red and green on each slide. So we're going to have 2 technical reps of each of these. How should we allocate these to slides? I don't know the answer. I'm going to show you one possibility just to show you how about thinking through it. Well, one thing that is quite simple to think about is, oh look, we've got 4 litters, 12 slides, 4 divides 12. Why don't we just do it like this and use the first 3 slides for the first litter and so on? Well, I have to fill that out because we've got the 2 colours on each slide. And so then what you might do in this first one, use 1 and 2 on that slide, 2 and 3 there, 3 and 1 there. And I've done it in that way to make sure that the treatment contrast is orthogonal to colours. Well, I can do the same thing in each litter. Now, normally if I just showed you this part of the design, you wouldn't know, for example, if you look at those 4 2s, you wouldn't know which ones come from the same animal. But because I've done it by litters, you know that the same animal has got to be like that. What sort of a mess does this produce? Horrible. I'm not going to show you all of this. Do you remember I said that the treatment contrasts within groups had some confounding of 1/3 with the litters? Now, I deliberately chose to make whole litters confounded with slides differences. Here is something where this part of the treatment information picks up slide-to-slide variability and litter-to-litter variability. Something we probably don't want to do. I will also just show you when you've got efficiency factors, say here, of treatments within, this is treatments within animals, within litters is 2/3, but they've got 1/4 there. You have to multiply the efficiency factors. So, is this sensible? We've picked up 2 large variances. You might take the point of view, there's only 1/3 of our information there, but hang on, there's well, there's some more there. I mean, that one comes up all over the place. You might say the only sensible information on this comes up here, but actually that's only 1/2 of your information. So, if you've got partial confounding, what should we do? We've got something partially confounded with large variance, but a fairly small efficiency factor. Should we take the lesson from before and try to go for a stratum with small variance? Or should we say, hang on, there's not much information there anyway, we're not going to do anything about it, so we may as well confound with something bigger. So I mean, that's just on an example. As you can imagine, I try several designs and work through it, and often with some of these small ones the answer is that everything you do is bad. I mean, but at least this way you can decide how to choose between them. So let's look a bit more generally. Suppose that in the first phase we have some treatment term that comes out in 2 strata. This variance is bigger than that one. And I've put P and Q for the efficiency factors so that you might think that they add up to 1, which is true. At the second phase, we think there are 2 strata which we might confound with. Obviously, the best thing is to put them both in the one with the smaller variance, but that's not always possible. Let's suppose, I mean this seems a little artificial, but I think it helps to think about it. We label them so that this stratum goes with that one at the next stage, this one goes with that. Now, eta 1 is bigger than eta 2. Should we match up big with big, or should we cross over the big to the small? Anyone know the answer? Okay, so this is just summarizing the thing. Well, this is not a hard result, but it's just to show I did prove something in my time at the Institute. Assuming if you know the ratios of these things in advance, then you can say what would be the right weighted least squares estimator, and you can find out the variances. You get the smaller variances by keeping big with big if Q over P is bigger than this thing. Otherwise, you should swap it round. What is this thing? I chose these so that eta 2 is smaller than eta 1. This number, therefore, is less than 1. So if Q over P is bigger than 1, we're certainly in this case. And that's the usual thing. We've got P to be rather small. So what this says, if we've got a small loss in phase 1, then you should go ahead and put the big there with the big there. But there's a rather grey area where you might have to actually evaluate things. Well, as you see, those of you who are being really sharp will see that the abstract I put up at the beginning had a word in that didn't have when I...
Are you combining information in that slide?
Yes, that is a combined information, and it's assuming you know the relative values in advance. So I'm not allowing for what may go on because of the tails of the estimation and so on. This is just but it gives some guidance, I think, if you know in advance. So sometimes, as we've seen in some examples this week, you're actually going to put in some more treatments at phase 2. Well, these are dead obvious principles. Well, I really enjoyed in Keith Baggerly's talk yesterday. Some of the things you have to say are so obvious you wonder why you have to say them. Plan it all in advance, especially if you want to estimate interactions between the phase 1 treatments and the phase 2 treatments. This is a potato storage example from Roger Payne that appeared in a paper of Chris's and mine. The first phase, we've got 4 cultivars and 3 fungicides. We're going to have a complete block design. Later on, we're going to take this stuff and store it on pallets in shelves in such a way that all combinations of cultivars and blocks go on shelves, fungicides go on here, and now we're going to have storage times as being the extra treatment things we put on. Well, you can do this by design key. You separate out 4-level things and 3-level things, and so long as you never mix up pseudo factors from the different levels, you can get the design out quite simply. So I'm going to alias C and F with plot factors. Then at the second stage, the appropriate thing for cultivars needs to go on shelves, blocks has to go on shelves. I'm pretending that pallets begins with Q because I couldn't think of another word for pallets, and then the times goes with that. And so you can write out the design and you get the ANOVA table very nicely out of that. Well, on Sunday evening, Terry and I went out to dinner. We had a nice South Indian meal, and he asked me a design question. I said, no, that's too hard, go and ask Angela and Sue. Well, of course, by Monday I had the answer, and I gave it to him, and he managed to show it in his talk on, Tuesday. That was only yesterday, wasn't it? Well, I thought I'd done enough, but then he threw me out a challenge in his talk, so I had to go away and incorporate this example. It's not just his fault, actually. It's Ching Shui Cheng and Carla Vivacqua as well, because some of the stuff they gave in their talks on Monday is also relevant to this. So, in case you've forgotten, this is the background. It's a bit simplified, but it's to show you the principles. Things that might affect the way you collect blood. Now, he was talking about doing it on 4 people, doing it 4 times per person, and just to simplify it, I'm cutting out the other factors that he had there. And so the piece of paper I gave him on Monday showed him how to do this design, 10 2-level factors to combinations of people and times, so that nothing is confounded with people. He said, well, maybe we can get 8 samples from each of these, and then we've got some processing factors and some storage factors. So I haven't actually consulted him on this, but I think some of these factors could be put to any old combinations here, but probably the storage factors have to be applied to whole batches at a time. So this is Terry's question to me, and I'm going to turn it into a question for you. What might we think about? We might think about confounding people with batches. But some of these storage factors are already going to have batch-to-batch variability. If we do that, we're going to add in the person-to-person variability for that. We might think about doing the sort of complete opposite and then make the people orthogonal to batches. But of course, in some sense, that wastes or uses up some more degrees of freedom. There is a compromise, and this is what showed up in Ching Shui Cheng's talk and also Carla Vivacqua's talk, where we could split the people into 2 lots of 2 and the batches into 2 lots of 4, and make this matrix with 2 empty boxes and split the things like that. Then you get this factor, which I write with this V sign, the supremum of the people factor and the batches factor. It's a factor with 2 levels, 1 there and 1 there. And as we saw in those talks earlier in the week, there is a single degree of freedom there, which you have to allow for. This single degree of freedom has person-to-person variability and batches-to-batches variability. So that's larger than all the other variances. There isn't any other degree of freedom there that has that variability. But on the other hand, if you can avoid confounding anything with that, that might be a good thing to do. So what I would tend to do at this stage is say, here are 3 strategies. Let's look at them and see what we could do and what happens there. So, okay, before I come on to the conclusions, this is something I would normally say here. Sometimes you can obviously say, this is the best design, go for it. Sometimes you could say, here's a design, they have their various disadvantages. And what I do at that stage is go back to the scientist and say, which of these disadvantages, do you think is worst? And quite often the scientist will say, oh, I was telling you a lie. I can easily get 8 more animals. But it's only by you pointing out that you've done your best and all the designs that you've got have got some disadvantages, and they realize that actually you could do something else. So I don't think it's up to us to make that final choice. You should go back and have that discussion. So what are my conclusions? As I say, these are things that I don't need to tell you, but I am telling you anyway. So I've talked specifically about 2-phase experiments, but we've seen earlier in the week that multi-stage batch reprocessing experiments work in a very similar way. Think about the whole thing right at the start. Think about small variance. Think about residual degrees of freedom. If you can be orthogonal to bad blocks in the first phase, try and keep orthogonal also to bad blocks in the second phase by confounding the boring things with each other. If something you want to know is confounded in the first phase with a large variance, try to put it with small variance in the second phase. On the other hand, if you've got partial confounding, then the best design depends on efficiency factors and some idea about the ratios of those stratum variances. Conclusions 2. These are just some conceptual things that I find help. I don't insist that people use them, they help me. These randomization diagrams that come from Chris Bryan. I haven't quite used them. I have these panels to show tiers of factors, and in complicated cases I've given up trying to put those arrows on because you get things like that red spider I showed you. I haven't talked about this, but I do use Hasse diagrams to work out the strata and their degrees of freedom. Jingshui Sheng and Chris Bryan showed you how to do that earlier in the week. If you still don't know how to do it, you can go out to CUP this afternoon and buy the book. The design key method of construction for orthogonal designs really helps you work through things in a systematic way to work out what's possible, and you can just go through it step by step and work out what's confounded where. And I find that these tables, skeleton ANOVA tables, really help me assess the qualities of the design, even though I might not be using ANOVA to do the analysis. So I hope there's lots of questions there for people to think about. Thank you.
So, as Rosemary said, are there questions?
Or answers. Angela?
Okay, so this is a question really. Back early on in the talk, when you were talking about the analysis and getting different answers which way round you put in your terms, so you're talking about mixed model analysis, which I'm not that familiar with. If it's a fixed effects analysis, then of course it's crucial which order you
Yes, of course.
So you don't have the same.
But the thing is, some of the software for mixed models, the way it fits the variance components is the same as the way it fits the fixed effects.
Yeah.
And that's why it matters. It shouldn't, but it does.
Yeah.
So if you've got a degree of freedom that comes in 2 terms, it will fit it in the first one.
Right.
And because it's doing that, it won't allow for the fact that, where you should be adding the 2 variances together.
Yes.
Ben, you have?
Yeah. You talked about ANOVA, so you're assuming a response on a continuous scale with usual...
Yes, Ben. I'm only thinking about a response on a continuous scale.
But one of your earlier examples, I think almost your first one, seemed to contemplate panels of testers at the end. So what relevance would this work have for choice experiments?
I'm not talking about choice. These are people who'll be grading on a scale, say 1 to 10, which, as other people have said earlier this morning, we often do treat as continuous. But yeah, I mean, how on earth does any of this generalize to, say, binary outcomes? No idea. Steve?
Where you had the ANOVA where you had different estimates of the same variance component, was the issue there was if you're testing the fixed effect, which estimate should you?
Yeah, I didn't. Normally I do this in such a way that I can jump back in large blocks. But I think...
The question I was going to ask was, would one of the choices you could make be equivalent or sort of analogous to what you would get from a pure randomization test? Sort of David Cox was mentioning.
Well, I actually don't know. I mean, in David Cox's talk on Monday, he didn't, I think, oh no, that's being rude. He talked about randomization tests and he talked about randomization justifying a model. They are not at all the same thing, which is why we have this different point of view on how to randomize Latin squares. And I'm getting this from a randomization model for each stage. If you were to do a randomization test, I honestly don't know what you would get. I'm just saying if you did a straightforward ANOVA on this, you would compare that with that. But in doing that, you're ignoring the fact that you, actually not that one, this one here, you're ignoring the fact that you could get some information on this from up there.
What I'm not clear about is what exactly you mean by a straightforward ANOVA.
Okay, you can get at the hypothesis testing stage, I would compare that mean square with that one. And when I give you the variance for the estimate of the difference between those, I would use that thing there multiplied by something. And perhaps I could get a better estimate of that thing there by using this information as well, which might need the Satterthwaite stuff.
Okay.
I don't know. Henry?
Can I ask a somewhat naive question? I mean, it seems that you have this choice between fixed effect, sometimes, not always, a choice between a fixed effect or random effect. You can't have both. There would be essentially aliased if you introduced a fixed effect where you already have a random effect to measure that.
It depends. It's true.
So I know it's a philosophical question. I'm never quite sure how you make the choice between whether you have a random effect or fixed effect.
Okay. I mean, the way I do it is different from what you heard from Chris yesterday. I'm saying here, I'm taking a randomization model at phase 1. So these things are random, and I'm doing a randomization at phase 2, so those are random. So my fixed effects only come from those treatments there. But we saw an example where, you might decide you're not doing a randomization model and something from a different panel might also be fixed. And when you do that, I mean, so ignore that for the moment. This sort of table still shows you clearly what's aliased with what. And of course, if you've got two fixed effects aliased with each other, you're absolutely stymied.
Yeah, that I understand.
Yeah. Oh, yeah, of course.
It's the aliasing between the random effects and the fixed effects which is confusing.
Hey, last one.
I think if I can interpret your question, Rosemary, it's that you can do your hypothesis testing in a sort of standard unadventurous way by using it as an ANOVA table. Then the issue is how you estimate stratum variances in order to combine information.
Yeah.
Now, clearly throwing it into a REML analysis will do that if you can manage to specify that correctly. We agree that there's a problem in the aliasing in some cases.
Yes, yeah.
What might be interesting to do, and this is analogous to the way in which hierarchical generalized linear models duplicate REML for the normal case, is somehow to take the information out of those sums of squares, then put it into a generalized linear model in order to, gamma log would be the thought, in order to estimate the log variance components, which would be analogous to the normal case. I think that's something we might talk about.
I think so, as I say, but unpicking the aliasing is why you can't just be simple-minded about it.
But you're fitting a linear model, so you can specify that in the context of a design matrix for the estimation of the dispersion parameters. So
Okay. Yeah, I'm with you. Yeah.
Okay, let's thank Rosemary again for excellent.